My name is Christina Sigurdsson. I'm a professor of pathology at UCSD. And it's really a great pleasure and honor to introduce our speaker today, Dr. David Eisenberg, who many of you know from his contributions to resolving amyloid structures. I've had the very good fortune to work with David on studies of prion protein aggregation. And David is a professor of chemistry and biochemistry and also biological chemistry in the medical school at UCLA, as well as a Howard Hughes investigator. And he's also the director of the UCLA Institute for Genomics and Proteomics. David he's truly a giant in the biochemistry field. Prior to investigating amyloid structure, he made many fundamental contributions in biochemistry, including the relationship between amino acid sequence and tertiary structure, identification of protein motifs, predicting protein-protein interactions. And more recently, in a series of landmark papers, his team reported on the fundamental structure of short key segments of amyloidogenic proteins, showing that within the spine of a fibril, amino acid side chains are interdigitating in a dry steric zipper down to a resolution of 2 to 3 angstroms. And his team published a whole series of studies on this work. First, the atomic structure of cross beta spines in the yeast prion protein, SUP35, which was published in Nature in 2005. And then the structures of 30 other amyloid spines from mammalian-derived proteins, published in Nature 2007. Followed that with studies of structure-based drug design for blocking fibril elongation. That was published in Nature 2011. And then Nature 2015, the structure of the toxic core of alpha-synuclein. Really a tremendous, tremendous series of achievements. So how did he arrive at this point in his career? He initially was studying biochemistry as an undergraduate at Harvard, where he was encouraged by protein chemist John Edsel to study basic science as a career instead of going into medicine. And so he did this. He was a Rhodes Scholar at Oxford, where he earned his PhD studying hydrogen bonding and ice and water with theoretical chemist Charles Coulson. He then moved to Princeton to work with Walter Kautzman as a postdoctoral fellow studying hydrogen bonding in water and ice and their thermodynamic properties. And he wrote a book together with Dr. Kautzman on the structure and properties of water, which I believe is still in print. He then continued his training at Caltech, where he worked together with Richard Dickerson on protein crystallography, and that was prior to joining the faculty then at UCLA. He's been extraordinarily productive. He has published over 300 papers and has numerous awards and honors, including membership in the National Academy of Sciences, the American Academy of Arts and Sciences, and the Institute of Medicine. And he's also won the Harvey Prize, which was given for pushing the technical limits of crystallography and elucidating the structure of amyloid fibrils. And so today he will be speaking to us on the latest work from his team on functional amyloid and how it is converted to pathogenic amyloid. Thank you so much for being with us today, David. It's really a pleasure to have you here. And just to let the audience know, this talk is being recorded. And also you can please post your questions in chat, or there will be opportunities to ask questions live at the end as well. So without further, yes.
Thank you so much for inviting me to speak at UCSD and for arranging meetings with you and other faculty members there, which I'm really looking forward to, and for giving me a rain check for the dinner that we're missing today.
Yes.
I'll just correct one of your exaggerations of your introduction. I'm the director of the UCLA DOE Institute. I passed on that job to others. So today I've got 3 topics. One is the structure of amyloid fibers, of pathogenic fibers, and what makes them so stable. And then how we go about inhibiting the formation of these fibers and the cell-to-cell transfer of the pattern that spreads from cells to cell, giving the example of tau, though we've worked with other proteins as well. And then the difference between functional amyloid, and we're seeing more and more examples of functional amyloid. In fact, more examples of functional amyloid than of pathogenic amyloid. And what are the differences in the structure and properties of functional and pathogenic amyloid, and how functional amyloid fibers can convert into pathogenic amyloid? So this is the work. Present, we have 16 members in the lab. They're listed at the top here, but they're preceded by 30 or more former members who worked on this since the late 1990s. And of course, we have a whole list of collaborators who made a lot of this progress possible. And then I have got disclosure at the bottom. There's a small company in Thousand Oaks, which is takes some of these discoveries and tries to make them into practical form. And I'm happy to have questions at any time, so please interrupt. I'm glad to have your interruptions. So I'm going to start with the structure of the so-called NAC core element of alpha-synuclein. It was established as essential for fiber formation by Virginia Lee and others in the 1990s. And a postdoc in our lab, Magdalena Ivanova, who's now professor at University of Michigan, she was able to grow crystals of it, but she didn't know, and we didn't know that she had grown crystals because they were so small you couldn't see them in the optical microscope. But then about 2015, Jose Rodriguez, who was then a postdoc in the lab, and others discovered that there were tiny crystals there, seeing them in the electron microscope. And Jose, who's now on the faculty of UCLA chemistry, he was able to determine the structure. And here is that structure, which illustrates the stability of amyloid fibers. There are always pairs of sheets, and the side chains interdigitate like the teeth of a zipper. And the stabilization comes from the tight mating of the sheets and the polarized hydrogen bonds. And now you're going to see the teeth of the zipper. It looks just like a zipper there, interdigitating. The hydrogen bonds go up and down the fiber. You can see them with the dashed lines here, and they're aligned above one another, so they're highly polarized. Quantum mechanical calculation shows that these hydrogen bonds are stronger than those of ice. There are also side chains in many of these structures that are stacked. I'm going to show them to you later. In this particular structure, we don't see those stacked side chains, but often tyrosine, phenylalanine, glutamine, and asparagine are stacked on the outside of these fibers. So this structure that I just showed you, I'm going to switch to a pointer that you can see here. The structure that I showed you up here in the upper right of this slide, we call that NAC core. And here's that structure I showed you. And if we look down the fiber axis and put the van der Waals radius on the atoms, you see they completely fill the space. It's extremely tightly packed. And this structure is seen at the core of the fibers which are formed by alpha-synuclein. So here are two structures of alpha-synuclein determined by Lin Jiang and Vincent Lee and David Boyer. And this one of these structures is this red one on the right. It's a twisting fiber. And if we just take a cross section, that's shown here, this cross section through the fiber, and you'll see there are two filaments, protofilaments, which twist around one another, forming the fiber. And each protofilament is a stack of layers, and each layer is one of these bent polypeptide chains of this core of alpha-synuclein. And they meet at this point here, and this point is the structure of NAC core. It's almost identical to the crystal structure of what is holding the two protofilaments tightly together in the core. There's also another tightly mating segment in this NAC region. We call that a pre-NAC. And again, the crystal structure was determined by Jose Rodriguez. Here it is. And again, you can see on the upper left that the space is completely filled by the atoms. Again, very, very tight. And that's at the core of another fiber structure that alpha-synuclein forms, another polymorph, we say. And again, the same sort of overall bent horseshoe, they meet at a different place, and that place in this fiber is at pre-NAC rather than NAC core. Both fibers are formed simply by a stack of these identical layers, but they meet at a different point. So now to give you a little review of the many structures that have now been determined by cryo-EM of amyloid fibers. We call these near atomic structures. The resolutions are in the range of 2 to 3 angstroms, where the crystal structures I just showed you, they're at 1.5 or better angstrom resolution, where we really see the atoms there. Here we don't really see the atoms, but we can trace the polypeptide chain and infer where the side chains are. So there are about 40 of these structures. I'm showing you half of them here in this slide. And the common theme is, first of all, that there are several polymorphs for each of these sequences. So this is alpha-synuclein. To date, I think there are 5 different polymorphic forms, same sequence, but it folds in 5 different ways. That is, forming layers which have different structures. Here, 3 of them, and then the layers in all of them are stacked up, as I showed you before. Some of these are from our lab, some from other labs. The Goddard lab in Cambridge, Shears and Goddard, have determined 9 structures for tau, different polymorphs. I'm going to show you those in more detail in a minute. From our lab, we've seen 4 different polymorphs of TDP-43. We can get some idea of the energy using what we call salvation energy. We calculate the energies, and stabilizing residues that tend to stabilize are shown here in red, destabilizing residues in blue. And here's the structure that I just showed you of alpha-synuclein, and you can see where the 2 protofilaments meet. They're very stabilizing residues, and that is the NAC core right at the junction of the 2 protofilaments. In 3 dimensions, that's what's shown here. And other residues can be destabilizing. So this destabilizing pair are a pair of charged residues on the surface, but they're oppositely charged, so they tend to compensate each other, and then they're bound water molecules probably, though we don't see them, stabilizing these stacked charges. And here's a very stabilizing residue. It's a tyrosine. It's stacked up. The rings, phenyl rings, are on top of one another, thousands of layers, giving stability to these fibers. And we see the same sort of energy relationship in these other structures. Now, in many of them, there are what we call zippers at the junctions of identical polypeptide chains. These are homotypic zippers, meaning that the 2 sequences which form the zipper are identical. And we see that in TDP-43 and in these other structures. So often when there are 2 filaments, it's the same sequence which zips together at the interface, giving a very stable interface. But we also see in these structures what we could call heterotypic zippers. And these heterotypic zippers, again, the side chains are zipped up, but the 2 sequences are different. And we see those in many of these structures in the interior of the structure. Inside the bent horseshoes, you see these heterotypic zippers. So the common features of these pathogenic fibers, each strand or protofilament is a stack of identical bent protein horseshoes or hairpins. Actually, the formal name for these is protein arches. Most fibers have 2 or more protofilaments. Some have only a single one, and sometimes there are more. We have one in which we see 27 protofilaments. These bent hairpins or arches are 2-dimensional structures, so they're different from globular proteins and membrane proteins, where they're 3-dimensional structures. But in amyloid, the polypeptide chain is essentially in 2 dimensions. So they're much simpler in a way than globular and membrane proteins. These stacked hairpins and arches tend to bond at homotypic zippers. So when there are 2 or more protofilaments, we see that the 2 protofilaments bind at these homotypic zippers, but they're stabilized each chain by heterotypic zippers as well, as I showed you. A common theme is they're multiple polymorphs, so the same sequence folds in different ways. And that's true in almost every case. And then there are stacks of charge residues, which are mainly solvent exposed. They're not in the interior, which would be destabilizing. They're outside the fibers and compensated by water and paired with oppositely charged stacks. Now I want to ask 2 questions. The first is, why are there so many polymorphs? For example, in tau, there are 9 different polymorphs, which are shown here. Actually, one of them is not really a polymorph. The Pick's disease protein, it's alternatively spliced. So the sequence is not exactly the same as the other 8 tau polymorphs, but the Goder lab has found these 8 other structures that the tau sequence forms, the same sequence, different structures. Why are there so many? Well, our hypothesis is that these structures don't have adaptive functions. So they haven't been selected. They just happen. And Anthonson's thermodynamic hypothesis doesn't apply. That is, evolution has not selected the sequence which always folds into the same structure. Of course, we need that for our other globular and membrane proteins. We can't have cytochrome C that folds into 9 different structures. We need the same structure in each cell. So it looks as though these amyloid polymorphs are not evolved structures. So that seems to be why they can fold into different ways. But now, why, when Goder and colleagues look at just Alzheimer's brain, do they only see these 2 polymorphs? Well, they look in their published papers, 17 different Alzheimer's brains give just these 2 polymorphs, so-called paired helical filaments. In CTE brains, they looked at 3, and it's a different polymorph, but always 3 are all the same. Pick's disease, 3 brains give the same polymorph. And so why is that? It's rather mysterious, isn't it? So again, we have a hypothesis on that, and that is maybe this particular polymorph, the paired helical filament, it causes the disease. Or another possibility is some cofactor, which could be the pH or some other molecule in a particular brain region. It causes Alzheimer's disease and also causes this polymorph. So there's somehow a particular polymorph or strain seems to be associated with each of these diseases. All right, so that concludes the first of my 3 topics. Be glad to take any questions on that before we move on, or we can move on to the second topic. Anyone want to comment or raise a question?
There is a question from Haiyang Yu. About alpha-synuclein amyloid, he asks, will the N and C terminal sequences 1 to 46 and 78 to 140 change their conformation after amyloid formation?
We just don't know what those conformations are because they're not part of the core structures of the fibers that can be seen. So they are, to some extent, disordered, possibly very disordered. So yes, they may change the conformations. They may be free to bind to other molecules that aren't in the preparations that we form to get uniform fibers for the structural studies. So we have very little information about them. All right, so I'm going to move on to second topic, which is inhibiting these amyloid fibers. And I'm going to start with a little movie here that illustrates the overall idea. So this is actually a crystal structure of a segment of tau. It's one of the adhesive segments of tau. And this is how we envisage fibers of tau forming the fiber. And you can see this dark gray region, that's the steric zipper. And you see that it's binding the fibers together. And in the case of tau, there are 3 different interfaces, not just that one, but 3 different ones we've seen in crystal structures. So all of these are crystal structures. Now, our inhibitor is red here. It's a peptid which binds to the top of the fiber, and it has side chains that prevent other tau molecules from adding. You could see there how frustrated those other tau molecules are. They can't add to the fiber once this red peptid, which has been designed from the structure, once it caps off the fiber. So I'm just going to show you a little bit more detail how we design these peptids. First of all, we predict which segment of tau is an adhesive segment. We have an algorithm for that. These red bars shows the part of the sequence that's likely to be an adhesive segment. And then we synthesize that segment. We grow crystals of it. We determine the atomic structure of the crystals. The crystals are almost all invariably small. So this shows a typical crystal where they're just micrometers. In the case of those alpha-synuclein crystals that I showed you, they were more in the nanometer range, a few hundred nanometers. Once we see the structure, now we can design a peptid which will bind to the tip of the fiber, but have side chains which protrude out and stop other tau molecules from adding to the top and to the sides. And so we've been able to design 9 different peptid inhibitors for tau. And then we test them in vitro, and we use Mark Diamond's biosensor cells. So these are cells that overexpress a fluorescently labeled tau. And what we do is extract fibers from the brain of Alzheimer's or other tauopathy patient, and then we seed the cells with these extracted tau fibers. And if the fibers, they go into the cell, and then they induce puncture formation. But if we first inhibit the fibers from the brain of the Alzheimer's patient with one of our peptid fibers, now we don't see the puncta. So I'm going to show you that. Here's an example of this work of postdoctoral fellow Paul Seidler. Here are fibers that have been extracted from an autopsy brain of an Alzheimer's patient. They always look the same. They are the paired helical filament structure I showed you before. And here they are added to the biosensor cells, and you see the puncta that form. The white area shows a cell that doesn't have puncta. If we incubate these fibers overnight with one of the inhibitors. This one we call WIW. All the sequences have been published and are available. If we incubate it with WIW, no puncta form. Another one of our inhibitors, W4, it's no good against Alzheimer's, the extracted fibers from Alzheimer's patients, and puncta 2 form. A third inhibitor, M4, that one is good again, like WIW, and it prevents formation of puncta. So this shows the 9 different inhibitors. Here's WIW, completely inhibits puncta formation. So does M4, but W4 does not. That's here. W4 does not inhibit.
David, this is Don Cleveland. Could you go back to that slide? I was confused. Actually, it's your title. You say that the efficacy of specific inhibitors depends on things like sex and age. Why would that be? Especially, if I understand correctly, you're using these diamond cells as the assay. Is that?
Well, yeah, we don't have many studies of sex, Don. We do have a couple of age. So we've only had about 20 brains samples, and what we find is that there can be differences in this profile, this sort of profile that I'm showing you here. Differences in the profile depending on the brain region, depending on age, and I think in one case we saw that a female patient was different from a male.
I see.
Patient.
Okay. Yeah. I mean,
so we really
N of 20 is for most structural things. That's a big number, but of course it's a small number in terms of total patients. Yeah.
Right.
Yeah. Anyway, thanks you.
Good question. And this is again the profile of the different 9 inhibitors. These 9 inhibitors have been designed on the basis of 5 different segment structures. These segments come from 2 regions of the tau sequence, the beginning of the 2nd and 3rd repeats. And what you see here is from different donors, these Alzheimer's donors, the CTE donor, PSP donor, CBD donor, that some of these peptide inhibitors, particularly M4, seems to inhibit almost all of them. WIW is also pretty good, but W4, which I showed you before, it inhibits only the formation of puncta from the CTE donor and not from any of the other donors. So we begin to see that different polymorphs, or you might say strains, respond differently to the different inhibitors, which makes sense because the interfaces are different. So that concludes my comments on inhibitors. And what I can say is we've designed inhibitors not only for tau, but also for amyloid beta. For P53, that's the only one that's been validated in animals. So P53 forms amyloid fibers in ovarian cancer, and it halts, the inhibitor halts the formation of fibers and seems to be effective in mice where the tumor cells have been implanted. That's the only one so far that's been in animals. So now I go to the last topic, my 3rd topic, which is these functional amyloid fibers and their conversion to pathogenic fibers, which I think is going to be a very important topic in the long run. We got into it through Steve McKnight, University of Southwestern Texas, University of Texas Southwestern, who had been working with the protein FUS used in sarcoma. It's a 526 residue protein, but the N-terminal domain is the so-called low complexity domain, in which there's a biased composition of amino acids. You see that there are many serines, glycines, glutamines here, but lots of other residues are missing of the 20 coded amino acids. And McKnight showed that this domain forms hydrogels and undergoes liquid-liquid phase separation. And he sent it to me and asked, could we learn anything about the structure of the hydrogel? So Michael Hughes fused it with M. Cherry, graduate student Michael Hughes, and he found that it formed a hydrogel. Here's the M. Cherry fused with this domain, and it forms a hydrogel in this 5 ml beaker. He inverted it, and the gel comes out on top. If he takes his pipette and sticks it in this hydrogel and stirs it, it goes back into a liquid and it goes back into the beaker. He puts it in the refrigerator overnight and then turns it over again. Out, it comes out as a gel. So it's a reversible gel. So then he diluted it by factor 400 times, and there were fibers there. I was interested in the fibers, and Michael Hughes then took the X-ray diffraction pattern, and I was surprised that these have the signature of amyloid, the 4.6 angstrom separation between the layers and the 10 angstrom separation of the sheets. But they seem to be amyloid-like, yet it was reversible, and all the amyloid fibers we've worked with before were highly irreversible. So Michael Hughes, very energetic young man, he looked at the sequence and he found that there were repeats like this SYS, GYS, and here we go, SY, SSYG. He found several of these and he put them into crystal screens, and sure enough, they crystallized, and he determined the structures. And I'm going to show you these structures. These structures, now they're just segments. They're 6 to 10 residue segments of the structure, and they're quite different from steric zippers. So here's a steric zipper from amyloid beta at the top, and if we look at just the backbone, it's the pleated sheet, Pauling's pleated sheet, and the two segments form these zipped up structures with interdigitating side chains, and this is a very stable structure. But in all of these structures that Michael Hughes determined from the low complexity domains, now the backbones are highly kinked, and they don't form these interdigitated structures. They're just flatter interfaces without side chain interdigitation. So these were the 5 that he determined at first, and he termed these LARCs, for low complexity amyloid-like reversible kink segments. So all of these are reversible as opposed to the structure at the top. So these LARCs are similar to steric zippers in some ways. They're many layered pairs of beta sheets. They're stabilized by tyrosine and phenylalanine ladders. You can see these ladder here on the surface with phenylalanine or tyrosine residues here stacked up on one another, giving stability to the structure. And the interfaces are dry. There's no water in them. But they also are distinctly different from steric zippers. Backbones are kinked. There's little side chain interdigitation. The area buried in the interface is relatively small and more polar. When we calculate the energies, they're a third to a half that of the steric zippers, and these structures are reversible. And now I have another movie which will give you a better picture of this difference between the LARCs are on the right, steric zippers on the left. You look down the steric zipper, you see the interdigitation. Down the LARCs, the side chains don't interdigitate. There's stacked tyrosines all over the place in this particular one. As the structure rotates, you'll see in the LARCs, there's nothing in the center. The black line is just showing you the fiber axis, but there's the zipper structure in this steric zipper. So the steric zippers are like glue, and the LARCs are more like Velcro. They come apart. So Michael Hughes then did what we call 3D profiling. I'm not going to go into detail, but what he did was to look in the whole human proteome for low complexity domains, and there are about 2,500. About a quarter of our proteins have low complexity domains. And then he looked at those for those that contain LARCs. And the ones which contain LARCs are the ones which are found in stress granules, P bodies, Cajal bodies, nuclear speckles, lysosomes, nucleolus, nuclear porins, and keratins. Those are the proteins which are known to form so-called membraneless organelles of liquid-liquid phase separation, and they all contain these LARCs, which we believe is probably the interaction which forms the fibers which contribute to the liquid phase separation. So again, looking at the energetics of these functional amyloids, here's a structure determined in our lab of HNR and PA2, one of these proteins. It's an RNA binding protein, which forms liquid-liquid phase separation. And what you see is there are very few stabilizing red residues that are holding the chain together. That's in contrast to this pathogenic amyloid, serum amyloid A. Look at this very stabilizing interaction here. And the same is true of the functional amyloid FUS, very few stabilizing residues. So these functional fibers are less stable than the pathogenic fibers. They also seem to be monomorphic as opposed to polymorphic. So no matter how many times Murray, Tico, McKnight form fibers of FUS, they form the same structure. They don't form other polymorphs. And Xiaowei Liu in our lab has formed fibers of this protein, HNR and PA2, several times. They form the same structure. So these seem to be evolved structures, monomorphic as opposed to polymorphic. Now, when we look at proteins like these two, FUS, HNR and PA2, and TDP43, we find that these proteins contain several steric zipper segments and also a bunch of LARC segments. And we found in one case that we've studied, TDP43, there is a LARC segment which has a known pathogenic mutation associated with the ALS category of diseases. And when we take that segment and we determine the structure, we see a LARCs in the wild type sequence, but in the pathogenic sequence, that disease-associated sequence, now when we form that structure, it's a steric zipper. And when we measure the stability of these two segment structures, the mutated one is stable. This shows increasing SDS, and it takes a larger amount of SDS to melt this structure than the wild type structure, which melts much more easily. So this looks like an example in which a known mutation, pathogenic mutation, converts LARCs to a steric zipper. And we have a few other examples from these proteins. So just to conclude here, to give our ideas about this, these proteins like TDP43, FUS, HNR and PA2, and HNR PA1, they contain both LARCs, which are shown here by blue V-type structures, and steric zippers, shown by these red lines. And these proteins, under functional conditions like stress conditions, they can form a reversible fiber, which is mediated by these LARCs rather than the steric zippers. But under other conditions, one is mutation, another is phosphorylation, then these LARCs can be converted to sequences which form steric zipper-like structures, and we get irreversible fibers rather than reversible ones. And still, there are other conditions like extremely high concentration. We can drive these proteins in, from reversible to irreversible conditions. So that's our thinking on this conversion. So just to summarize here, there seem to be two classes of amyloid fibers. They're the pathogenic class, which we see in, trying to get my pointer here. There we go. There's the pathogenic class, which we see tau, amyloid beta, alpha-synuclein, and others, and the reversible amyloid type fibers, which we see in these other proteins, FOS, TDP43, and they can be converted to pathogenic form by mutation or other conditions. The origin of the pathogenic amyloid seems to be sporadic, perhaps mediated with cofactors. That's under investigation in a bunch of labs. The reversible amyloid structures seem to be evolved. The adhesive interfaces are mediated by LARCs and reversible amyloid structures, steric zippers in the pathogenic amyloid. The energies for the pathogenic amyloid are about three times or so that of the reversible interactions. The pathogenic amyloid seem to be polymorphic. They've not been evolved. It depends on the particular conditions what structure forms. If my brother is unlucky and his tau forms into the paired helical filaments, he gets Alzheimer's disease. If I'm lucky, my tau aggregates in my brain, but it goes into one of the other structures. I'm lucky and I don't get the disease. The reversible amyloid structures seem to be monomorphic, a single evolved structure. And we're able to make designed inhibitors for both pathogenic and also for reversible amyloid. We're experimenting with different sorts of inhibitors for these two different things. I won't get into that today. Just to show you the photographs of some of the scientists who've done this work. Paul Seidler has done the work on tau, including the inhibitors, and he has just received an appointment as assistant professor at the University of Southern California. David Boyer is an advanced graduate student. He's an expert in cryo-EM. He's determined about 10 cryo-EM structures from these various proteins. Kevin Murray is a wonderful MD PhD student who's worked on FUS and HNR and PA2. Jiawei Liu, graduate student, determined the structure of HNR and PA2. Funding comes from Howard Hughes and National Institute of Aging. So I thank you for your attention, and I'd be delighted if you have further questions.
Thank you so much, David, for that really very impressive seminar. There's a couple of questions in chat I can read. One is from Bill Mobley. How do we pursue a possible cellular cofactor for the Alzheimer's disease polymorphs?
Yeah, let me just expand, Christine, if I could expand.
Yeah, go
briefly.
ahead.
So just to thank David really for terrific insight. And David, this is mostly a question focused on how now we link these really different kinds of structures to the cell biology and to the brain biology of these conditions. And so specifically with respect to AD, do we have the tools, do we have a way of looking at these different polymorphs in the context of cell function or structure?
Bill, excellent questions. Just beginning. In the case of the GO-DARE lab, in two of these structures, CTE and CBD, they do see molecules within the structure, but the resolution they're working, they cannot identify what those molecules are. They can infer the side chains to which they are bound, and that gives them an idea of the charge of the molecule, nature of either hydrophobic or hydrophilic, and whether it's positively or negatively charged, but nothing beyond that. We are trying to isolate fibers from patient brains and then doing mass spec to see if we can identify the bound molecules. So that might be one way of moving from structure to cell biology.
Yeah, let me just continue on. So if the biologists were to have access to one or more stable polymorphs, let's say one polymorph, and we're able to not just look for it in the cell, but actually to treat cells with that polymorph, one could begin to think about really this underlying cell biology of what that polymorph does, assuming we knew where to put it, when to put it there, and what to measure.
Good point. The only way we know of getting a stable polymorph is to isolate it from brain, because when we take a recombinantly expressed protein and form a fiber, it's a different polymorph.
Yeah, that's what I was afraid of.
But maybe if we can identify appropriate cofactors, then it could become possible to make the appropriate polymorph in vitro, which could be used then in your experiment.
Yeah, and David, Don Cleveland again. So you're arguing these cofactors are actually assembled into the final structure. Is that?
That seems to be the case, Don, in CBD and CEV. What do I mean? The athlete's disease. In both
CTE, yes, right.
Thank you.
Yeah,
in
thank you.
both of those, they can see some molecule. They are not post-translational modifications because there's no covalent bond. They can see it's separated at a non-covalent distance. So it's a bound molecule and not something that's been covalently bound.
Okay, so you mentioned early on, and it was probably for, we old folks, with Anfinsen and the idea that things would go to a natural low energy state. And I still am, as one of the premier structural folks, are you now really comfortable with the idea that you can form a whole bunch of different kinds of very stable structures, maybe in different brain regions because of different cofactors or because of other proteins that happen to be present there? It just seems to me, and I remember seeing Mark Diamond make a bunch of different tau forms, or argue that he had done so, and that it just seemed, and I really both liked it, and I just found it very perplexing that there really could be so many different stable forms, and your data, just amplified on that with more precision. Are you like me still really surprised at that, or is it just that I drifted away from structural biology so long ago that I just can't really capture the new the new reality?
I'd answer that, Don, by saying we're also used to Anfinsen's ideas. It's really the central pillar of protein science, that a given sequence folds into a single given structure. Everything we do is built around that. But that doesn't seem to be the case with these amyloid polymorphs, that there are multiple structures that can form. And it depends on some extent of kinetics. So I'm not comfortable with it, but I think the facts support that. And if you look at Anfinsen's Nobel Prize acceptance speech, was in 1972 in Science. It's really beautifully written. And he says in that his he calls it the thermodynamic hypothesis. It holds only for proteins in their natural environment. I think he calls it natural milieu. The rule holds only for the environment in which they've been evolved. He also says in that talk, which is quite startling, back in 1972, that he believes that there must be other molecules that aid in the folding, that even though in vitro his molecules fold in a single way, that in cells it's the responsibility of other molecules that are helping. So he anticipated chaperones.
Thank you.
He really was a very brilliant scientist.
There's a question from Steve Olson. Why are the inhibitors pre-incubated? Do the inhibitors work without pre-incubation?
It depends on the inhibitor, Steve. So to give them the best chance, we inhibit it overnight. If we put them in and then with the fibers and then immediately apply them to the biosensor cells, they seem to work not quite as well. There'll be some punctus. So there's some kinetics of forming the interaction of the inhibitor with the fiber.
I had a question about fibril stability. Are these fibrils, do you think, likely to be fragmenting readily in the environment of the brain, or are they so stable that it's more likely that the oligomers and the smaller fragments that we're seeing coming from small aggregates that are just starting to assemble? And a second question on that, on stability question, can you predict stability by the conformation of the fibril? Is there any correlation between the fibril pitch and stability, or do the rod and twisted polymorphs have differences in stability?
The first question on fragmentation, which was studied by Weissman and Tanaka and others, the fibers, the more stable the fiber they found, the more readily it fragments. Fragmentation probably goes on. It may be that in younger individuals with fragmentation and chaperones, fiber formation is not a serious problem. Fibers can be dismantled, but as we lose chaperone activity, as we age, that even though there is fragmentation, we're not able to get rid of the fibers. That's pure speculation, Christina. And then in terms of energy calculations, I think our energy calculations are quite approximate. I think all energy calculations are quite approximate, including ours. We can't at present use the Rosetta programs, for example, because these structures, the cryo-EM structures, are not extremely accurate. So there will be, in the final models, there will be some overlap of atoms, of non-covalently bound atoms, so that if you now calculate the Rosetta energy, it's just huge and unrealistic. So we use our salvation energy calculations, which avoid an overlap term, and I don't think they're very accurate, and I don't think they could distinguish between two polymorphs of alphas and nuclein. I think they're good enough to distinguish between the two different types of amyloid fibers, but I won't go too much further than that.
Okay, thank you. Are there any other questions for Dr. Eisenberg? Okay. Well, thank you very much.
I think there's a question from Dr. Galasco
Oh, okay.
in the chat.
Yes. Okay, question from Dr. Glasgow. Do the inhibitors share structure with cellular chaperones, and do natural chaperones inhibit fibril formation or elongation?
Our inhibitors do not share the structure with chaperones. Chaperones do, they are able to dismantle fibers. There are some nice studies on sub-35 fibers by Tanaka in Japan. He has a series of papers, and the chaperones can actually take apart fibers quite readily. We are experimenting with other types of inhibitors other than peptides, which I think may be much better for delivery to the brain, and I have high hopes for some of these.
Okay, if there's no other questions, I'd like to thank David for this absolutely fantastic seminar and for sharing your work with us this morning. Thank you so much.
Thank you, Christina, for inviting me in this virtual visit to San Diego.